Okay, on to our next DNA process. We're going to get into DNA replication. So we're going to talk about the mechanisms of DNA replication, where and when it occurs. We'll talk about the concept of semi-conservative replication, and then we'll also look at some of the important roles and important components for DNA replication, and then how DNA kind of gets shortened in the process of DNA replication. So DNA synthesis, sometimes called DNA replication, basically has to happen before a cell divides into two, right? So we start with 46 chromosomes, and we're going to have to replicate all of those so that when we divide, each cell will still have 46. So we basically go from 46 to 92, and then in the process of division, go back down to 46. Now, this replication process has to happen during the S phase of interphase. It is not going to happen while the cell is dividing, right? Because we want to make sure that we replicate everything before we even think of dividing. The enzyme that's going to help with this process is called DNA polymerase. Similar to RNA polymerase, right? We talked about that in the last video. DNA polymerase has the exact same type of job. Basically, take that DNA and create a second copy of it. Now in this case, we're going to go from one single chromosome, and we're going to copy it start to finish and join them together at what's called the centromere, so that we have two copies that are 100% identical to each other of that same chromosome. Now, before a cell can replicate, we have to replicate the DNA. So the cell cannot divide unless we have replicated the DNA first. And so we have to go through what we call nuclear division before we can go through cellular division. And replicating that DNA has to happen before we do the nuclear division too. So we have a process, right? DNA replication, nuclear division, and then cellular division. So nuclear division basically has 4 basic steps, and you probably know these steps better as mitosis. Basically prophase, metaphase, anaphase, and telophase. But mitosis is not the only way to divide the nuclear materials, right? We can also divide to create gametes. So mitosis is used for like making more cells, like if you get hurt, right? Or you're growing, right? You think of like when a little kid is growing, they're getting taller, they're getting bigger. Or if you get a scratch, right? I have little foster kittens right now at time of recording, and they have teeny tiny little needle claws. That means I'm going to get little scritches and scratches from them. And so mitosis is used then to replace the cells lost and damaged, basically from those little scratches. So we can replace the cells that are lost. Meiosis is going to be used for gamete formation. So that's going to be used to produce things like eggs and sperm. Now, which of the following are highly mitotic? Basically, we want to ask which of these are going to go through mitosis rapidly and often. So embryo or a fetus, a neuron, an intestinal epithelial cell, and cancer. So some of these are going to be more mitotic than others. Something like an embryo or a fetus is going to be highly mitotic because we're doing a lot of growing and a lot of development in a very short period of time as an embryo. I know anyone who is pregnant might not feel like that it's a short period of time, but it is in the grand scheme of things. This is also why certain medications are not safe for a developing fetus, even though they're perfectly fine and harmless for an adult, right? It's because as an adult, you're not going through a highly mitotic process, whereas an embryo is. A neuron, nope, not highly mitotic. We're not dividing very often with the neurons. Same with the intestinal epithelial cells. We'll replace them as needed. It'll be more than the neuron, but it's not going to be on the same level as the fetus or embryo. And then cancer. Cancer is going to be highly mitotic. We've basically lost control of the mitotic process with cancer, so that cells are reproducing way faster and way more often than they absolutely need to. But we'll get into mitosis in the next video, so let's go ahead and focus on DNA synthesis first. Now, DNA synthesis uses a process that we call semi-conservative, right? The replication process is semi-conservative. What this means is that when we use the DNA to make a new DNA molecule, we're going to conserve one of the original strands and use it as a template to build the second strand next to it. So basically, we start with one DNA molecule that has two old strands. We pull the two strands apart from each other and then use the old strands on each of those to build a new one. So when we look at the new DNA molecules, each new DNA molecule will have one new strand and one old strand. And there's some really cool experiments that were done to figure this out. We're not going to get into that details, but if you have questions or you're curious, by all means you can ask and I will talk you through that. But let's go ahead and talk about how we get there, right? Because it's not just as simple as pull the strands apart and build a new one. We have mechanisms in place to actually do this. So similar kind of steps we saw with like transcription, right? Initiation, elongation, and termination. So initiation, there's the two strands of DNA get separated, right? Because it's all together, right? Twisted up together. And so we have an enzyme called helicase that kind of in a way unzips the two strands of DNA. It's kind of like if you were to unzip a zipper, right? When the zipper is zipped up, right, the teeth of the zipper are twisted and pushed together in a way that makes them inseparable. But then as you use the zipper down, right, you are separating the teeth, and so you create the two strands. We then get to elongation, where we have a DNA polymerase to bring the complementary nucleotides, to use the old strand as a template and bring the complementary bases to build the second strand across. And then termination, once we finish the two original strands, like we have the original strands unzipped all the way, we've built the new strands on both, then we stop replication because there's nothing left to replicate. We've already gone from start to finish. And that's a key thing that I didn't talk about in the last video, but I'll talk about now, is that the DNA expression, when we express a gene, we're only creating RNA for a very small section, right? Just like a gene. Whereas DNA synthesis, we're doing the whole molecule, right? From start to finish, cover to cover. But at the very ends, it turns out that when we have linear chromosomes like humans do, when we go through this process, we can't actually get the very last little bit, right? There's a little cap at the very end that we just can't quite replicate because there's nowhere for certain primers to attach. So every time we replicate, we get a little bit shorter, right? Not a lot, just a little bit shorter, a few bases or so shorter every single time we replicate. Now the good thing is that our chromosomes have sections at the ends called telomeres. And these telomeres are meant to be chopped up, right? They're meant to be lost in this process. So there's nothing important on those telomeres in terms of genetic information. It's just a section of DNA that we can afford to lose. But these telomeres shorten over time, and eventually they get too short and we can no longer replicate without destroying important information. And so that's when we are going to stop the cell cycle, when these telomeres get too short. The cell cycle will actually stop, will enter senescence, where the cell basically just stays in the current state and it doesn't divide anymore. Now, there are some cells that can divide indefinitely, like cancer cells, because they have an enzyme called telomerase. And this telomerase enzyme basically will keep re-lengthening that telomere over and over again. So the telomeres never shorten, and so we never get that signal to stop the cell cycle. So in this case, that sounds like a good thing, like, oh, we would not have to enter this senescence phase. But this means that we have lost control over when to stop a cell from dividing indefinitely or infinitely, right? We don't want cells to lose that control. We want to have a stopping point, especially because as cells get older, right, they can acquire damage, they can acquire changes that will get passed on if the cell divides. And that's important because DNA replication is not perfect. There will be mistakes that are made. Some of these are just luck, right? They just happen. Others are because you've been exposed to a chemical or UV light or something that will generate a mistake. These mistakes can result in mutations. Now, we do have pathways to help find mistakes and correct them. So like how you would have spell check, right? When you write a paper, you can hit spell check, it'll go through, it'll look, and it'll find all the spelling mistakes that it can and point them out to you so that you can correct them. And so you can find a lot of mistakes that way, but you can't find every mistake. So for example, if you misuse the word R, let's say you meant to use the word R, O-U-R, but you spelled it A-R-E, spell check will not be able to catch that. So some things will get through, and those are things that end up as mutations. Now, mutations that happen in your somatic cells, so this is what in like your tissues, like your skin or your stomach or your muscles, basically after you're born, you're not going to pass those on to your babies, right? You're not going to pass them on to your offspring. But if it's a mutation that happens in your germ cells or in your gametes when you make sperm and eggs, those can be passed down to offspring.